Chapter 4, Section 3. This is on proteins. This is the second of our macromolecules. And here are some familiar sources of protein in our diet: fish, steak, chicken, bird, and eggs. Okay, proteins are the workhorses of cells. They do lots of different things for us. Perhaps one of the most commonly known functions of proteins is they build muscles for movement. So muscle is actually unique to animals, and all animals have muscles, including Arnold Schwarzenegger and in the movie Conan. One of the best movies ever, but this is not a family-friendly film. So watch it by yourself or with some friends, but no kids around. Proteins are also used in our immune system. Things like antibodies. They're used to defend ourselves against pathogens, and those antibodies are made up of proteins. And they also help our cells recognize each other along with the different types of carbohydrates. And of course, proteins are very important for carrying out chemical reactions. They're called enzymes. And in this case, this is an enzyme that is breaking down sucrose into glucose and fructose. So this is an example of a catabolic reaction. Enzymes can also be anabolic, where they build other molecules up. Here's an example of an enzyme in action. This is actual computer models of proteins working as enzymes. And these proteins, they can be incredibly complex. So all proteins are made of amino acids. Amino acids are the building blocks of proteins. So what that means is a large protein is made beginning with a sequence of amino acids strung together. So what you're seeing here, glycine, valine, proline, alanine, those are individual amino acids, the building blocks strung together. Now, what you're seeing is also what is called the primary sequence of a protein. So every single protein will have a primary sequence of amino acids. We also call this a polypeptide. Poly means many. Peptides have to do with the amino acids being held together by peptide bonds. Here's an example of a polypeptide. Remember, all proteins are polypeptides, but the difference between a protein and a polypeptide is that proteins are folded into specific shape. So this molecule right here has a very specific shape. It has a primary structure of the sequence of amino acids, and what you're looking at here is the tertiary structure. And we're not going to get into tertiary, secondary, and quaternary structure. Just know that it's folded into the correct position and it has biological function. So this is a polypeptide, and specifically this is a protein because a polypeptide that's a protein has been folded. So all proteins are polypeptides. But not all polypeptides are proteins, because a polypeptide could be nothing more than just a sequence of amino acids. Here's a very large tumor-suppressing protein. One of the things about proteins, they're incredibly varied. There are potentially 1 million different proteins in the world, all made up of the same 20 amino acids, and they may have thousands of amino acids making them. And this is, like I said, a very large protein right here. This is the general form of an amino acid. And the best way to learn this is to draw it. And amino acids are the building blocks of proteins. They're made up of the amine group, a carboxyl group, and lastly the R group. This is the side chain. R is not an element. It stands for something that's there. So when I say that there are 20 different amino acids, there are 20 different side chains. Okay, so proteins are polymers. That means they're built up by condensation reactions. So here's our 2 amino acids, and when you put them together, water comes out, and you've linked the 2 amino acids with what's called a peptide bond. And the peptide bond is, you can see the arrow, it's right between that carbon and the nitrogen. Peptide bond is a covalent bond, but it's the specific name of a covalent bond between 2 amino acids. And I now have what is called a dipeptide. Di just means 2. Peptide means that I basically have 2 amino acids. And when you get a polypeptide, you have lots and lots of amino acids strung together. Now, if you build a protein by condensation reaction, you break it down by hydrolysis reactions. Okay, and this is condensation reactions. This is how we build the primary structure of a protein. And then there you can see it again with the individual amino acids. So peptide bonds are formed between amino acids forming the primary structure. And because peptide bonds are covalent bonds, these are strong bonds. Okay, we're going to skip secondary structure. We're not going to worry about that here. You'll learn more about that if you take more classes in chemistry or in biochemistry. But basically the tertiary structure, all proteins have this. This is basically when the protein gets folded together. And this is important because one of the underlying themes in biology is shape matters. So for these proteins to function, they have to be folded into the correct shape. If they're not folded in the correct shape, then they may not function very well. Now that being said, some proteins might exist in one of several different shapes, and those shapes might be something like an on or an off shape. Okay, let's go back to amino acids for a second. Here we've got two different types, alanine and serine. One is hydrophobic, one is hydrophilic. Now I know you're looking at that amino acid going, I know that carboxyl is hydrophilic and that amine group is hydrophilic. And you are correct. And they're hydrophilic because those can form hydrogen bonds with water. However, whenever we classify an amino acid as hydrophobic or hydrophilic, we're not worried about that backbone. And the backbone is that NCC, the amine group and the carboxyl group. No, when we classify an amino acid as hydrophobic or hydrophilic, we only care about the side chain. Now instead of R, I'm actually showing real side chains. So hydrophobic is alanine, CH3. That's just carbon and hydrogen. Those two elements have the same electronegativities. Those are non-polar covalent bonds. They are incapable of interacting with water. They can't form hydrogen bonds with water. Serine. I basically take alanine, add a hydroxyl group to that methyl. Guess what? Oxygen, hydrogen. Oxygen, way more electronegative than hydrogen. As a result, you get polar covalent bonds. And if you have polar covalent bonds, you can form hydrogen bonds with water, and you are now considered to be hydrophilic. Okay, you may have heard of essential amino acids. Now, we require 20 different types of amino acids to make all the tens of thousands of proteins in our body. So the essential amino acids, there are 9 of them. There they are. We have to have them, but we can't make them. So we have to get them from the foods that we eat. So good sources of all your essential amino acids, which we call complete protein. If you like to be a vegetarian, which is nothing wrong with that, you have to be careful with your diet to make sure you get everything you need. But quinoa is a great source of proteins. And if you do eat meat and eggs, eggs are also an excellent source of protein. Eggs are very digestible by our body, and the proteins are, if we break them down through hydrolysis reactions, then all the amino acids become readily available to us. All plants have all 20 amino acids. The problem is that some plant material may have so few of the amino acids we need that it can be hard for our body to digest them. But a healthy vegetable sources with spinach, kale, broccoli, and legumes, and mixed with different types of rice will give you all the amino acids you need. All proteins must be in the right shape for them to work. So if they lose that shape, they no longer work. So when a protein loses its shape and loses biological function, we say they become denatured. Now this is a big deal. And one way you can denature a protein is by adding heat. So if you've ever taken an egg and thrown it in a frying pan and scrambled it, the proteins in the egg have become denatured and permanently denatured. So what that means is whenever you cool the eggs back down, they don't go back to looking like the original egg. They're changed forever. Now that has a lot of significance to us. We can cook fish. If you've ever marinated a piece of salmon in lemon juice, the lower pH also changes the shape of the proteins in the fish and make it look cooked. So change in temperature, especially heat, and change in pH, these things can cause proteins to denature, which is where they change shape and lose biological function. Now this is really bad for us if we get overheated. Humans operate around 98 to 100 degrees. We can get a little bit hotter than that. But if we become overheated and our temperature gets up 105, 6, 7, 8, we're in big trouble. And the reason why is because our proteins will begin to denature and we can cause permanent damage to our bodies, especially in our brain. So think about this. Every emotion you've ever had, every thought you've ever had, every memory you've ever formed, all of that is stored in proteins in your brain. It's all about biochemistry. So when you become overheated, not only can you damage the organs in your body, you can damage your brain and cause permanent brain damage if you become overheated. And the problem is, the reason why it's permanent is because once you cool down, those proteins may not go back to the normal shape.